Microbial metabolism, part 1. Metabolism means the sum of all chemical reactions that are occurring inside of a cell. We have some metabolic reactions that are catabolic, or the word catabolism, which means the breakdown of large molecules into smaller molecules with the release of some form of chemical energy that's useful to do some type of work or have some kind of action performed. The other kinds of metabolic pathways that are occurring inside of a cell are the anabolic, or what we call anabolism. That's where we take the smaller molecules plus the energy that's available and make large molecules. Since we have these two types of biochemical reactions occurring inside of a cell, both catabolism and anabolism, we call metabolic pathways amphibolic. The amphi prefix means two. So we have two pathways, or two different types of pathways, generally occurring almost at the same time, where we have the breakdown or the catabolism of large molecules to small ones with the release of chemical energy, might even be some heat energy, and then we have anabolism using the small molecules and the potential free energy in the form of chemical energy or heat energy into larger molecules. Now, the word anabolism, there's another word that means essentially the same thing, and that's biosynthesis. So anabolic pathways, biosynthetic pathways, are the same kinds of metabolic pathways. For catabolism, another word you could think of, when we're breaking down large molecules into smaller ones, think of decomposition. That's another word that could mean essentially the same thing. In most classes that discuss metabolism, the general example would be carbohydrate metabolism, where we talk about glucose and the breakdown of glucose, and then what do we use with the simple small molecules and energy that are produced from glucose breakdown. But we want to make certain that we also understand that there are other kinds of foods, in addition to carbohydrates, that can be broken down into small molecules that can then be used as building blocks in biosynthesis or anabolism to make new pieces and parts of cells. So in addition to our discussion about carbohydrate metabolism, we will also have a very small discussion about fats and proteins and how we can break those complex foods down into small functional building blocks or molecules that can then be used in the biosynthesis of other materials that cells need for their structures. In our discussion of metabolism, we have to mention oxidation and reduction. Frequently, in metabolic biochemical pathways and reactions, oxidation, or the loss of an electron and the loss of a hydrogen, is coupled with reduction. Some other molecule gains the electron and gains the hydrogen. These are often called redox reactions and are very commonly observed inside of cells in a variety of biochemical metabolic processes. As we work through our examples of metabolic reactions and pathways, we will see examples of redox reactions in a variety of areas. The easiest way to remember which is to remember the little saying, Leo says Gur. Leo, L-E-O, loss of electrons means oxidation. The Gur, gain of electrons, means reduction. These kinds of reactions are coupled or paired together. And if you take a look at this illustration, you can see that certain types of molecules will gain the electrons and the hydrogens, and the other kinds of opposite sides of the reactions is where we see the loss of the electrons and hydrogens. So the coupled or paired reduction and oxidation reactions that are often found in metabolic pathways. In our metabolic reactions and pathways, we're going to need a biological catalyst in the form of an enzyme to facilitate the changes of our starting materials, which we call substrates, into what we call end products. Now, in a typical chemical reaction, we often use the word reactant to mean the starting materials, and then we use the word product when we're describing what's produced in the chemical reaction. With an enzyme catalyzed reaction, we change the word reactant to substrate, and we modify the word product to end product. When we use those specific kinds of terms, then we automatically know this is an enzyme catalyzed reaction. Notice that when we're using an enzyme, the enzyme is going to essentially combine with the substrate or the starting materials. We will have an intermediate structure that's called the enzyme-substrate complex, and it's at that point that chemical bonds are either formed or broken in the changing of the substrate into the end product. Enzymes generally have a very high degree of specificity for their substrate. In other words, the enzymes won't react with just any kind of starting materials. They're very specific for their starting materials or substrates. Now, if you take a look at the illustration on this slide, you'll see on the left-hand side there's an example of an enzyme that's in sort of lilac or light purple. And then we have two pieces of substrate in a golden color on the bottom. On the enzyme, there are almost like little puzzle piece places for the substrates to lock in place in the enzyme, and that's part of how the enzyme helps to facilitate the formation of chemical bonds, or if we were doing essentially the opposite, breaking chemical bonds. Notice that the two pieces of substrate fit nicely into the enzyme-substrate complex. The enzyme-substrate complex brings the two pieces of substrate close together, and then the enzyme falls away, and we have the end product of that newly formed structure that has been put together in this chemical reaction. Notice that the enzyme participates in the chemical reaction but is not changed itself. The only entity in the chemical reaction that changes here is the substrate. Enzyme participates but is not changed and is able to start again and some new pieces of substrate if needed and if possible. The characteristics described on the previous slide, enzymes participating in chemical reactions, that they can participate in multiple chemical reactions, that they participate and are not permanently changed, so that what we start with in the form of an enzyme, we wind up with the same enzyme. We also find that in addition to the previous characteristics described, enzymes are going to allow chemical reactions to occur in cells or in biological systems with less energy required. With less energy required, it's going to result in faster reactions occurring. And we find that if we want to describe what's happening and why the chemical reactions are occurring faster and with less energy required, we typically define or say that enzymes lower the energy of activation. This graph represents an illustration of what we mean by energy of activation. In a typical chemical reaction where we have reactants turning into products, notice that the very high curve on this graph is the illustration of the energy of activation needed without an enzyme to convert the reactants into products. At the very top of the curve, where we will see the transition from reactants to products, is actually called the transition state. And so, just like rolling a rock uphill, notice it takes quite a bit of energy to change the reactants into products, to get up and over that essentially energy barrier. With an enzyme-catalyzed reaction, the amount of energy needed to convert substrates into end products is much lower. So it's much easier to roll that stone or that rock up over that hill to get from substrates to products. Much lower energy needed to get through that transition state from substrate to end product. And that's what enzymes will do. And of course, this allows the reactions to occur much more quickly than if we had no enzyme facilitating or helping that chemical reaction to occur. In addition to the active site, where substrate would lock into place on an enzyme, we have two additional participants that might be included in the overall structure of an enzyme. One is called a cofactor, another is called a coenzyme. Cofactors are derived from minerals, things like zinc or magnesium. Coenzymes are derived from vitamins. And so it's part of why you're always told to make sure you get plenty of vitamins and minerals in your diet, because those coenzymes and cofactors, if you'll notice, are part of the construction of the active site of an enzyme. If your body doesn't have sufficient minerals and vitamins for the construction of the active sites of an enzyme, then that enzyme's not going to work at its best. In addition to looking at the coenzymes and cofactors that can be part of the construction of the active site of an enzyme, notice also that the remaining structure of the enzyme, called the apoenzyme, surrounding the location of the cofactor and coenzyme, is much larger. It's sort of the overall structure of the enzyme. The apoenzyme is typically made from protein. Now, not all enzymes are protein in construction, but most of them are. There's actually a tiny, tiny percentage of enzymes that we find in nature that are made from RNA, or ribonucleic acid. But this is a very small group of biological catalysts in the form of RNA. Most enzymes are protein in structure, and the apoenzyme component that's illustrated on this picture is what we're talking about when we talk about apoenzyme. It's the protein scaffolding to which the substrate can bind in the active site, and the cofactors and coenzymes might be needed in the construction of that active site. When we have all the parts together, the apoenzyme protein scaffolding, the cofactor and coenzymes if they're needed, then the whole entire completely functional enzyme that remains or that's been built is called the holoenzyme. Now, there are lots of ways that we can control enzyme function. One possible way is essentially a competitive inhibitor, a mimic, a molecule that has part of the structure of the original substrate, but it is not the substrate. So if we take a look at the image on this slide, the competitive inhibitor has a partial portion of its structure that's essentially the same kind of shape as the original normal substrate. If this particular competitive inhibitor attaches itself to the enzyme first, then normal substrate can't possibly bind to the active sites. If we can't bind substrate to the enzyme, then we don't make any end product. Another example of an inhibitor that would bind to an alternative site. So competitive inhibitors often bind to active sites, as in the previous slide illustration. On this slide, notice that we have some alternate binding sites that are called allosteric sites. The allosteric sites are distant from the active site. They're not associated with the active site at all. There's some other location on the holoenzyme, often the apoenzyme structure. And notice in this example we have what's called an allosteric inhibitor. The allosteric inhibitor, again like puzzle pieces, fits nicely into the allosteric binding site on the enzyme, and when the two combine, notice the illustration on the right-hand side of the slide, we've changed the shape of the active site by attaching this inhibitor at a distant site to the allosteric site, we have what's called allosteric inhibition. We've changed the active site, so now it cannot function when any type of substrate, when its normal substrate comes around, the normal substrate just cannot fit until that allosteric inhibitor is removed. In addition to allosteric inhibitors, there are some allosteric activators that can be found in nature. Just take a look at that illustration of the chemical reaction and imagine it's going the other direction. If we have an allosteric activator attached to the allosteric site and we have sort of a modified active site that substrate cannot bind, and go the opposite direction so that when the allosteric activator falls off, we've now modified the active site so that the substrate can bind to the enzyme. So we find that there are, in fact, ways of controlling enzymes with either inhibitors or activators and a variety of other ways. In addition to using activators and inhibitors to control enzyme function, we can also use temperature and pH. For temperature and pH control of enzyme function, we're talking about protein enzymes. It's not quite so effective on the few small percentages of RNA enzymes. But for protein enzymes, changing temperature and changing pH can have a dramatic effect. If you take a look at the temperature graph, notice that as we increase the temperature from the temperature minimum, which on this illustration on the graph is about minus 10 degrees Celsius, and we increase the temperature to roughly about 35 to 40 degrees Celsius, we have essentially gone from a minimum temperature that's suitable for function to the optimum, which is the best possible temperature for that enzyme. Now, as we increase the temperature a little bit more, from the temperature optimum to the temperature maximum, which is about 50 degrees for this enzyme, what's happening to the enzyme is we're essentially cooking it. We call it denaturing the enzyme because it's a protein in structure. When we denature a protein, we've modified the structure sufficiently and it's irreversible. We cannot go backwards and simply cool that enzyme down and have it essentially in the same shape and structure that it used to be, or that it was originally. The typical illustration we always give students about denaturing a protein is simply cooking egg white. Egg white in its natural state, when it's just out of the bird, is a gel-like material that's clear. When we heat that egg white, and it turns from clear gel to a solid white matrix, we've cooked it or we've denatured it. We can't take that cooked egg white, cool it down in the refrigerator, and have it turn back into that clear gel. So we always say that denaturing a protein, either any other kind of protein or if it's an enzyme protein, then it's an irreversible change to that protein. Once you've cooked an enzyme or denatured it, you've essentially destroyed it. It's no longer able to function. That's the other reason that we see on the curve that it's lopsided. The curve slowly increases when we go from minimum to maximum, but notice that we go from the optimum to the maximum. It's almost like falling down a cliff. We have a dramatic decrease in the function of that enzyme as we increase the temperature. Going from temperature minimum to optimum, there's a general rule that we often use when describing how temperature from minimum to optimum actually enhances enzyme function. We generally say for every 10 degree increase in temperature, we double the reaction rate of an enzyme catalyzed reaction. Now, the alternative to controlling enzyme function is pH, acidity or alkalinity. The pH range, just like the temperature range, goes from a minimum, the most acidic that we could use, to an optimum, which is the ideal, to a maximum. Notice that the bell-shaped curve for the pH range is much more symmetrical. because typically changing pH does not typically denature a protein. So we'll find that there is an optimum pH where we have ideal function, and then on both sides of that optimum, we can have essentially a more acidic or a more alkaline environment where the enzyme may or may not function.